Okay, let's get started. Welcome to this lecture. First of all, I'll take you on a journey through a topic that I really find fascinating and that is at the core of research done in my lab. I'll talk about how we might repair the damaged brain. And this is a theme that really gained strong attention and saw a lot of developments after the discovery that the adult mammalian brain has stem cells which constantly generate new neurons, and of course is a theme of relevance for medical advances in the way we treat neurodegenerative disease and brain injury. First, I'll share the outline of this lecture. I will start with the problem itself, so the impact of brain disease, and after a short introduction on neural stem cells and adult neurogenesis, we'll go through the possible solutions that have been explored so far and which aim at achieving replacement of neurons. And this is important to point out because the approaches I will discuss have the specific aim of replacing neurons that are lost during the progression of disease or in acute injury, and thus rebuild neuronal circuits. So there are other strategies like, for example, disease-modifying interventions that aim to stop or slow down the natural course of the disease and not to reverse, like is the case of neuronal replacement. So finally, we'll wrap up and have our Q&A session on Friday in the designated time, but I'm also very happy to take questions today in case you have already some. So here is the problem. You can see here in the picture a brain of a patient of Alzheimer's disease next to a healthy brain. And what is obvious is that the Alzheimer's disease brain in there's an obvious damage, there's atrophy, scarring, and I could show some similar pictures of other diseases like Parkinson's disease, Huntington's disease, and multiple sclerosis, where they all tell the same story. So collectively, these brain diseases represent one of the major public health threats of our times, and the numbers are increasing. We have at the moment 35 million people suffering with these devastating conditions. We all know somebody in our family or immediate cycle of friends that is affected. So indeed, the medical need is clear, and it was only a few decades ago that scientists started ventured on this quest. And that was partially because for a long time there was a central dogma in neurobiology, which was established a century ago by Santiago Ramon and Cajal, that in adult centers the nerve paths are something fixed, ended, immutable. Everything may die, nothing may be regenerated. It is key for the science of the future to change, if possible, this harsh decree. So this conclusion was based on Cajal's observations in histological samples of the mouse brain and depicted in many of his drawings, showing very complex and elaborate architectures, like you see some here, and which he called a full-grown forest, impenetrable, tangled jungle of nerve cells, where no new stations or lines can be created. But he also acknowledged that this view goes against the concept that we all hold of an organ that allows learning, formation of memory, adaptation to the environment, and thus an organ that must be, to some extent, amenable to change. And in fact, he posed a challenge to the next generations of scientists, basically to prove him wrong. And it was about 80 years later, in the 90s, that this dogma fell, with definite proof of the existence of stem cells in regions of the adult mouse brain. And those we can see in green, and this was also observed in most of the other species that were mammals that were studied. These stem cells have the ability to generate new neurons constantly. They do sit in two confined regions of the mouse brain. The one is the subventricular zone, And here stem cells generate neurons that migrate and integrate in the olfactory bulb, whereas the dentogyrus stem cells within the hippocampus produces local neurons. So this is also the case in the infant human brain, while neurogenesis in the adult human brain remains a topic under controversy, and studies also suggest a different placement of new neurons here in the striatum. So it turns out that the adult brain is penetrable after all, and incorporates new neurons throughout life. And of course, this raised a lot of expectations for neuronal replacement and circuit repair. However, adult neurogenesis is a privilege of only a couple of regions in the brain, as I just showed. But the regions affected by most neurological diseases are non-neurogenic, as it's the case for the cerebral cortex. So the fundamental question was whether it can occur outside of these areas, and thus with relevance for repair. And it was only 3 to 4 years later that a study in Lund was the first, and the first of many, demonstrating that these niches of neurostem cells are activated by injuries to the brain, and that part of the progeny ectopically migrates to the core of the injury. And just a word of caution here, that although the title of the paper claims neuronal replacement, what the authors and many followers observed was actually increased proliferation and migration, so very early stages of neuronal development, but did not probe whether those cells integrate in the circuits and thus replace the ones lost. And this is one example of such spontaneous response on the right side. In green, this staining labels the young neurons on their way towards the stroke site, which is very close to the subventricular zone, the neurogenic niche, and seen also in the sagittal scheme just here. So many of these cells are also birth dated by BRDU incorporation in red, and some already show very mature morphologies, as seen here in this inset. So this is not specific to injuries in the striatum, but just a pose which is really close to the subventricular zone stem cells, but also a similar response occurs when the injury is in the cerebral cortex, with neuroblasts forming streams oriented dorsally from the subventricular zone towards the cortex where the traumatic brain injury was inflicted. And here are examples of some of these cells with their typical migratory morphology. Now the stem cell niche, the other stem cell niche, the dentogyrus, also expands their progeny in an extent that seems to be dependent on the severity of the injury, in this case a stroke. So here we have mild, moderate, and severe strokes inducing different levels of neurogenesis. So, but contrary to subventricular zone progeny, here these cells do not migrate long distances, but instead are believed to settle locally in the dentogyrus. Now, a myriad of studies have come to these very same observations in rodents, and just a few years later, cells labeled for immature neuronal markers were also detected in post-mortem samples of a stroke patient, here in green and magenta, closely associated to blood vessels of the peri-infarct region, here in red and filled with erythrocytes. So these findings were quite exciting, since they indicated a kind of a self-repair attempt upon damage to our brain. But neuronal development doesn't stop in these early steps of migration. And further studies analyze the potential to differentiate in the right cell type and integrate into existing circuits. And several studies showed the acquisition of a neuronal phenotype that matched the site where the cells were, but others, like in the hippocampus, reported an abnormal differentiation and integration in circuits, which potentially contributed to malfunction of the circuitry. So now, a limitation across all studies was the low survival rate of these cells, which also limited long-term studies on their synaptic integration. So in fact, in mouse models of stroke, for example, 80% of the new neurons were estimated to die in the first 6 weeks. So these are recruited neurons. And only 0.2% of the dead striatal cells is replaced by recruited cells at this time point. So there were enormous efforts in that decade to understand the mechanism of progenitor cell activation and also recruitment to the injury, and use the knowledge to promote this phenomenon. So namely to stimulate cell proliferation, migration, and survival. And that coincided with the time when I started my PhD in 2006, and when I decided to dig into this burning question. So, and what we have described was a mechanism by which subventricular zone neuroblasts, here in red, invade the injured striatum by using blood vessels, here labeled in green, as a physical scaffold for their migration, as well as seen in this inset. So our time-lapse imaging in brain slices showed that they use two modes of migration, either a continuum along the blood vessel where the cell body sits, or by extending the leading process towards another vessel and basically jumping in between vessels, which both together enable dispersion through the striatums, through the injury site. And this is how neuroblasts in the ischemic striatum looks like, with very low dynamics. Most of the time is in stationary phase, but seldomly initiates a saltatory jump. Now, we have seen that vessels were not only a structure that these cells attach to and that they use to guide their leading process, but also express and secrete BDNF, which is a neurotrophin that is known to promote their entry into migratory phase. And that's exactly what we see here when we apply BDNF to the brain slices, that we stimulate migration of these cells. So this process is highly regulated and goes much beyond BDNF. Stroke actually induces microglial activation, astrocytosis, leukocyte infiltration into the ischemic areas, and all these cells produce numerous factors, including other growth factors, cytokines, chemokines, which have been also associated to processes of differentiation, of proliferation and migration of these cells. For instance, factors such as monocyte chemoattractant protein 1 and stromal-derived factor 1 are secreted at sites of inflammation and form a concentration gradient of attractive signals to these neuronal progenitors. So although the molecular composition of injury sites is rather complex, several studies succeeded to promote injury-induced neurogenesis. However, eventually the outcome is still negligible compared to amount of dead cells. So to summarize this first part, what I told you was that neuronal recruitment from stem cell regions of the brain to sites of injury is rather a universal response across acute brain injuries, which differs from the situation in chronic progressive and degenerative disorders, where often the effect is the contrary, with lower levels of neurogenesis and these cells stay confined to the niche. The spontaneous response is initiated by a multitude of molecular factors secreted at the injury, mostly by glial cells, which then engage stem cells into proliferation and attract neuroblasts that leave their niche and migrate attached to blood vessels. And as always, there are many open questions, but I will abbreviate to those that I find the most relevant in this subtopic. It is still a matter of debate whether the recruited neurons become the cell type needed. First of all, this depends on where the injury is inflicted, or if they stick to their normal developmental trajectory and become GABAergic granule neurons in a different place. So that is very unclear. And the question of whether the low survival rate is due only to the injury environment that these cells develop in and cannot cope with, or also an inability to integrate into ectopic circuits due to a cellular identity that doesn't match. So this strategy has been quite on standby, or let's say it has been at slow pace, developing at slow pace, because other approaches have been indeed showing much more promising outcomes. And I'll go through the two strategies that are indeed at the forefront of research in brain repair. So the second approach, which has really been blossoming, is the transplantation of neuronal progenitors in affected areas for neuronal replacement and reconstruction of circuitries. Progenitor cells for transplantation are obtained from either fetal brain or from expandable sources such as ESCs or iPSCs, and the cortical transplants that have been used in models of traumatic brain injury and cortical stroke. So cortical transplants have been used in these two models in rodents, but also striatal transplants have been performed in mouse models of and rat models of Parkinson's and Huntington's disease, which both ultimately affect this region. So both regions and most brain regions affected by damage or disease are indeed non-neurogenic. They do not normally accommodate neurons in their circuits during adulthood. So we answered to this question in the very first studies, like other labs, whether synaptic integration can occur in such regions. And here I need to make a small parenthesis, since transplantation in Parkinson's disease and cortical injuries have very different purpose. So stem cell-based therapy has actually been performed in Parkinson's disease patients, and last year new clinical trials have started again with the improved stem cell products and patient selection. So in this case, the transplantation aims not at reconstructing circuits, but at restoring dopamine transmission. So in the normal brain, dopaminergic neurons of the substantia nigra innervate the striatum, where they release dopamine. And dopamine plays a major role in motor control. Now, in Parkinson's disease, these cells start dying and the striatum becomes gradually denervated and devoid of dopamine. So transplants in this case are not placed where the cells die, but in the projection target, the striatum. And they aim, as I said, to restore dopamine transmission, not to reestablish circuits. So these cells will not be able to get the inputs, their normal inputs, and be regulated by the circuitry. They will work as dopamine pumps and restore the levels of dopamine. Now, on the contrary, in cortical injuries, the cells are plugged into the injury core for small injuries or next to the cavity left by large injuries, with the purpose of replacing neurons and repairing these circuits by rebuilding the full repertoire of afferents and efferents with the right regions. So this is the ultimate aim in this case. But can they do it? Can they integrate correctly in pre-existing circuits? So this was a question that I tackled during my postdoc in Magdalena Goetz lab, and therefore I'll tell you a little bit more about these studies. We transplanted reporter-labeled fetal neurons in the injured visual cortex of the mouse brain. The transplant basically fills a large part of the cortical depth, and cells remain in the transplantation site at the injury, as seen here in one section, but also in consecutive sections without much migration. And early on, the graft is composed of young neuroblasts, as revealed by double cortin staining, and they project away from the graft, which we can appreciate here, numerous and long neurites, towards subcortical structures, also presenting growth cones in their tips, which indicates an active growth. So axonal growth was not thought to be possible in the adult brain, and several studies have now, with transplantation, have now shown that that the adult brain is indeed permissive to axonal growth. Now, 1 month after transplantation, when the cells have acquired their final morphology, and I'll show you more on that, they express the mature neuronal marker NOIAN and also markers of cortical identity, as for example here CAX1. So these neurons molecularly resemble the local cells, and their developmental trajectory and also their tempo is as if they were developing in utero, which we did time-lapse imaging and could follow the development of apical dendrites and basal dendrites over time. And indeed, they follow very closely the brain development. But do these cells establish synaptic connections with the host brain? So this video shows the complex morphologies that they develop over time. And perhaps you can appreciate a pyramidal cell-like morphology with an apical dendrite and extensive arbors of basal dendrites. In these basal dendrites, we can visualize spines, and in these axons, which actually project to several millimeters away from the cell bodies, there are axonal button, so both structures that are correlates of synapses. So we next ask with whom these cells connect in the host brain, and to trace their connectivity, their input connectivity, we used a retrograde monosynotic rabies virus and observed abundant local inputs here in green surrounding the red graft, but also a broad innervation brain-wide from many different cortical and subcortical regions. So we showed that not only the connections were all normal afferents for that region, but also at a quantitative level, they closely resembled the normal circuits, with strikingly similar connectivity ratios. So the structural skeleton seems to be restored. Next, we ask if the circuits are functional. Do they process visual information coming from the retina? And one of those regions that innervates the transplant is the thalamic lateral geniculate nucleus, which is known to relay the visual stimulus from the retina. So this bottom-up visual pathway is now regenerated, and we asked if it is functional. And to answer this question, we performed in vivo calcium imaging during visual stimulation, and we showed that transplanted neurons respond to visual stimulus, here presented in gray periods. And over time, later at 15 weeks post-transplantation, these responses are tuned to a particular feature of the visual stimulus, which is a characteristic of neurons in the primary visual cortex. So this was very encouraging. And curiously, it seems to be a property of the injured brain only, as the intact brain is unable to connect or scarcely does so. So these findings showed that an injury is necessary to prime the environment for connectivity between host and graft. Now, when we compare the connectivity of transplants placed in various different conditions, from brain trauma to Alzheimer's mouse models, we came to the conclusion that there are obvious quantitative differences. So all these connections are normal with no aberrant inputs, but one mild injury leads to an extremely specific integration with a connectome that closely matches the old, also at the quantitative level, while others develop excessive number of connections with the graft. So the environment of the diseased tissue is itself a key and deterministic factor in neuronal integration. So what we know so far is that, as shown by optogenetic stimulation of these circuits or sensory stimulation, in combination with calcium imaging, that transplanted neurons can reestablish functional input circuits and also project to correct targets in the brain, and that the disease context, so the cellular and molecular milieu where the cells are placed, strongly influences the degree of connectivity they'll develop. And we have indicators that this is due to the presence of molecules associated with processes of synapse elimination. So, although we have been learning a lot about the potential of stem cell transplants to restore circuits, we still don't understand if transplants restore cortical microcircuits. In other words, if all the neuronal subtypes of the cortex are represented, or at least the main in the microcircuits, and if they establish specific connectivity patterns for a given neuronal subtype. Also, whether the long-range connectivity matches the neuronal subtype identity. So finally, a question that I also find fascinating is how axonal guidance and pathfinding is regulated, how these axons know where's the target and to whom to synapse with, and whether these new axonal projections are functional. This has not been investigated. And also, what is the importance of other cellular elements like interneurons, glial cells, and endothelial cells? And for example, glial cells are very well known to have a strong impact in synapse and synapse elimination. So now I will move on to the third approach that has been used for brain repair, and that is the youngest strategy to bring new cells into the damaged brain. And it consists of generating neurons from other cells in situ by reprogramming their cellular identity. So you are probably aware of these two pluripotent sources from which we can derive new neurons, and that are in fact used for cell therapy. So the ESCs, embryonic stem cells from the inner mass of the blastocyst, and induced pluripotent stem cells generated often from fibroblasts by applying the combination of Yamanaka factors, which reverts a somatic cell into a pluripotent stem cell state. Now, using a technology conceptually similar of cell fate conversion, it is possible to convert a somatic cell directly into a neuron without passing through an iPS cell state. By forcing expression of key neuronal transcription factors, and that has been done successfully using different starter cells, among which some of them reside in the adult brain, like astrocytes and pericytes. So astrocytes are particularly attractive as a cell population to convert, since these cells proliferate after an injury, and therefore their main astrocytic functions might still be covered after a fraction of them is converted to neurons. So astrocyte to neuron conversion was first done in the Petri dish, and you can see on both conditions, left and right, the alteration of morphology once I play the video, from a more astrocytic-like morphology, more star-shaped and flat, to a more neuronal morphology, since in both conditions MASH1, a main neuronal fate determinant, is expressed. However, there's an important difference that I will explain once I play the video. So you can, I think it's clear from very early on that these conditions are a little bit different and generate at the end of the video a different neuronal complexity. So it became very clear in the field of direct neuronal reprogramming, that this process imposes barriers that limit the efficiency of conversion. So that has been leading to the development of improved protocols that combine the induction of the transcription factor or transcription factors with, for example, expression of pro-survival genes like BCL2, and in these cases, much better results have been obtained. And this, as I said, it's in vitro. But in recent years, different groups try to convert parenchymal astrocytes or other local cells in the brain that around the injury and that can presumably be spared in an injury context into neurons of interest. And I will show here where the field stands by providing a couple of examples, so from a couple of studies that I consider to be the most significant achievements. So here you see a population of astrocytes that was targeted by a control virus expressing only a reporter gene. And you can see the clear protoplasmatic morphology of cortical astrocytes, while in the animals injected with a virus of the same serotype, but expressing neurogenic factors, neurogenin-2 and NUR1, together with GFP, the cells have now become neurons, as suggested by their morphology, already at low magnification, but also showed by staining for neuronal markers. And in fact, reprogrammed neurons, also called induced neurons, acquired a pyramidal morphology that is strikingly similar to normal cortical projection neurons. They develop spines in their dendrites over time and show electrophysiological properties of mature excitatory neurons. And that development is gradual, and as you see in the first time point, there are less spines, and yes, we don't have here the early responses, but several parameters were tested at the 2 time points, showing in all of them a gradual development of these properties. So at 2-3 months post induction of the transcription factors is when the cells seem to reach a stage that is compatible with the normal projection neuron. And at this stage, induced neurons also project to different areas of the brain. And remember, this used to be a cell that has a local territory of a few micrometers only, and now projects to the contralateral hemisphere and to subcortical targets, which are several millimeters away. And in addition, they also receive long-range inputs, which were revealed by injecting a retrograde tracer in the contralateral hemisphere after reprogramming and observing tracer positive here in fluorogold reprogrammed neurons. So it seems that also this strategy generates bona fide cortical neurons that resume the normal connectivity pattern of local neurons, but for behavioral improvement, new circuits need to be functional circuits that operate as the old circuits. And this was a major advance in the field. In this case, an astrocyte to neuron reprogramming was done in mouse model of Parkinson's disease, and specific fate determinants of dopaminergic neurons were used. And the authors show, yes, they validate that at molecular and electrophysiological level, they do resemble dopaminergic neurons, but most importantly, they demonstrate reversal of motor symptoms. So these are a few motor tests that where diseased animals, here in gray, show ipsilateral, for example, here show ipsilateral rotations. This is expected in this model. It looks this is in blue, so the naive, the blue bar shows the diseased animal with increased number of rotations. Now the diseased animals injected with the control virus expressing only GFP behaves exactly the same way as the lesion. While after expression of the transcription factors to convert these cells, astrocytes, into neurons, the animals show an appreciable improvement in behavior, and that is in all the different tests done. This is the same profile. So where the purple bar with reprogramming resembles the normal mice. So this table now summarizes the main aspects that need to be considered for a reprogramming protocol. So examples of paradigms used in experiments mentioned before, in vitro and in vivo. Some studies use human cells, and of course these are highlighted here. Of course, these are in vitro studies, while mouse studies use injections of different viruses, different expression systems, different starter cells as well. So it has been a field with a lot of variants of a protocol, and it has been without doubt a very hot field, given the advantage of using own cells for new neurons and avoiding the invasive nature of transplantations. So, and because of that, it is also a field that is moving at an impressive pace. So it took only 15 years from the first evidence of neuronal reprogramming in mice until the recent evidence of showing reversal of motor deficits with this approach. And of course, this strategy needs to go through all the adequate controls that the transplantation field already has gone through in several decades of research. And also, I think also interdisciplinary approaches between virologists, circuit biologists, and developmental experts will be very important to move this field forward. This approach, as it has been done so far, is also invasive to a lesser extent than transplantation because it implies injection of the virus intracerebrally at the moment. But there are efforts to perform neuronal reprogramming using AAV vectors that cross the blood-brain barrier and can be targeted to the cells of interest. So where are we in this field? So the reprogramming approach is at the stage where efficiency of reprogramming and acquisition of the right subtype of interest is well underway. Induced neurons acquire molecular and electrophysiological responses, also connectivity patterns similar to the normal neurons, and where the local neurons, let's say. And they were shown to improve symptoms in a mouse model of Parkinson's disease. So we know now more about this process of reprogramming itself from transcriptomic data, which provided also new tools to foster cell conversion. So namely to supporting a metabolic shift from glycosis to OXPHOS. So astrocytes rely more on glycolysis, while neurons are known to use OXPHOS processes. So there is a lot of cell death in this process of metabolic change that can be helped by promoting the OXPHOS metabolism. And where I think the field will see the next developments is in the understanding of molecular programs that are driven by specific sets of transcription factors in diverse source cells. So whether reprogramming events are largely shared by various donor cells or rather cell type specific, exploring epigenetic barriers to direct reprogramming, and also understanding the impact of tissue environment and injury signals. And with that, I get to the end of my lecture. I would like to thank my lab, my collaborators, of course the VBC services, and you for your attention. And I'm very happy to take questions if there are any now.